Who doesn't want anything created at your fingertips? I bet that would be refreshing. Using 3D printers, people can use plastics to make a variety of conformations, like figurines, masks, and even more 3D printers. With our current technology, we can create new skin to help burn victims, and we are getting ever closer to creating functional organs that could help those that need transplants. But what if we could combine these together to create live structures at our fingertips? Hi folks, my name is Cole, and I have a Master's of Immunology. Today on Investigate, Explore, Discover, we're going to be looking at live 3D printable plant material. So hang around with me throughout this whole video to get all of the relevant background information so we can dive into some exciting experimental results. There's also more information for you in the description below. Now, wood is and has been integral to how our society functions. It builds and furnishes our homes. We use it to cook our food and is the basis of so many of our daily products. However, to harvest wood, we need to cut down entire trees that take decades to grow, and there's a lot of waste associated with it. Not to mention the environmental costs of transporting wood to processing plants, processing it, and moving it into our homes. I'm certain that you remember how pricey lumber was during the height of the COVID-19 pandemic. Demand for wood is forecast to continue growing, and identifying a way to meet this while simultaneously mitigating the classical harms of current forestry practices is of critical importance. Some say that specific tissue engineering could be the answer. Tissue engineering is a complex interdisciplinary field requiring knowledge on the impacts of numerous interactive factors on growth and development of individual cells and tissues, and is controlled by three major factors: the cells used, the scaffolds to create structures, and the biological factors used to influence cell fate. Currently, the field has been primarily focused on animal cells. This has resulted in the creation of many fantastic advancements, like artificial skin for burn victims, and the generation of powerful models that enable studying of human disease and tissue function. This has also led to the advent of cellular agriculture, which creates selectively generated meat products not requiring the sacrifice of animals. Based on current projections, it is poised to be more cost and environmentally friendly. But by shifting this focus to plant cells, the same principles of non-sacrificial plant harvesting and reduced costs can be applied. Choosing what type of cellular scaffolds to use in tissue engineering is important, because shaping or constraining the physical limits of the growth environment influences macroscopic features of the resulting material. Now, scaffold-guided growth enables the production of materials both at scales and forms that are not naturally occurring, and can be scaled up to high-throughput manufacturing outputs. Bioprinting and casting techniques using hydrogel enable such processes and permits both freestanding and contained arrangements. Ideally, that means that plant cells can be grown in the shape of the desired furniture or building supplies. So think about how cool it would be to get kits to grow your own furniture, or even use Japanese woodworking concepts to craft larger pieces. Different parts of plants have variable characteristics and functions, leading to distinct material level properties. For example, cells in leaves are flatter, softer, and have more dermal tissue because their main job is to capture energy from the sun. Cells in the stem, however, have harder cell walls full of lignin because their job is to support the plant and enable cell growth. To do this, they are full of vascular tissues, like xylem and phloem, to transport water and nutrients between the roots and leaves. In trees, this is the tissue that is most precious because it is sturdy and readily usable. This manifests as differentiated cells exhibiting unique secondary wall patterning due to lignin, which indicates the transition to vascular cell types. This is important to consider when creating lab-grown plant materials because tuning the cellular level characteristics is also expected to be critical for controlling emergent material properties. Recently, using a common zinnia flower model, researchers found that plant cells can be controlled to grow in certain fashions. Depending on the nutrients used in culture media, like hormones or macronutrients, it can cause cells to selectively differentiate. These differentiated cells have different physical and chemical properties, and these attributes can be assessed through multitudes of different tests. dynamic mechanical analysis enables the non-destructive assessment of mechanical properties. This is done by holding the test sample between two fixtures, one stationary and another that imposes a time-dependent sinusoidal strain. The resulting time-dependent stress is simultaneously monitored by quantifying the torque generated on the stationary fixture. Stresses in phase relate to elastic materials, while stresses out of phase relate to solid materials. Wood, because there's a large variety, falls in between these extremes. And of course, these properties can change depending on whether the material is hydrated or not. Now, I want to take a moment and really highlight why investigating and understanding the growth and material properties of lab-grown plant materials is so important. Forestry practices cannot keep up with the growing need for plant materials, and growing plant material in a lab can supplement and eventually replace this gap. By understanding the developmental pathways for plant cell differentiation, we can customize material being grown to suit real-world applications, ultimately enabling us to cultivate materials at forms and scales that are not readily available in nature, making manufacturing and processing more efficient. Now, if you also think that these are some important reasons to research this topic, go ahead and tap the like button. This brings us to the paper that we're focusing on today. This paper is called Physical, Mechanical, and Microstructural Characterization of Novel 3D Printed Tunable Lab-Grown Plant Materials Generated from Zinnia elegans Cell Cultures by Beckwith et al. From MIT in Cambridge, Massachusetts, USA. In this paper, the authors provided new insights on cellular level tunability, culture scale and form control, and the physical, mechanical, and microstructural characteristics of engineered plant cells. When cells are growing, media formulations can impact cell fate, and to start their experiments, the authors first isolated cells from zinnia leaves and grew them in media for about 10 days. The first variables that the authors investigated was what affects the addition of varying levels of the growth hormones alpha-naphthalene acid and benzylaminopurine had on cells in culture. They tested cell media with very little levels of the hormones, and media with high levels of the hormones, and really everywhere in between. Cell growth characteristics were quantified using the fraction of live cells, cellular lignification, enlargement, and elongation. When these zinnia cells were cultured, they found that the low hormone growth media, also called maintenance media, caused cells to increase by nearly 80% and produce the greatest cell enlargement, while the high growth hormone media, also called induction media, showed a reduction in live cells, but increased lignification. The authors compared this new data with data they previously published 6 months prior, and found that the results were comparable in pretty much every regard, indicating that these results are replicable. Though to be truly replicable, they need to be confirmed by another group. Either way, to test how well scaffold-guided growth functions, the authors printed cells in various shapes and left them to grow even longer, for 3 months. They started with a tree-shaped structure in homage of what they hoped to replace, and found that the cells grew and differentiated into cells that produced lignin. Furthermore, just making the cells into an effigy of what they wanted was not the sole factor that determines the cell's growth. Other shapes could be grown too. Interestingly, when the samples were dehydrated after growing, an indicator of cell growth was the darkening of the scaffold gel. Now, the authors next wanted to compare cell growth from natural plants and the 3D printed scaffolds. They did this by imaging cross-sections of the grown material. You can see here an image of the cells in the naturally growing zinnia plant after 30 days. But this is very little when compared to the amount of cells present from the 3D printed grown material. What I think would be an interesting comparison, though, is the gross number of cells that are present in each sample, to get a true side-by-side comparison. Now, translation to larger scales requires cell survival in thicker structures. To investigate survival at greater culture dimensions, cell cultures were plated at various gel depths and allowed to grow for 32 days. The authors covered the cells with 2.5, 5, and 8.7 millimeters of gel to simulate different thicknesses. Personally, I thought it would have been more interesting to see cell-laden gels at these thicknesses. Either way, when the cells were assessed to see how well they grew, the authors found that the differing gel thicknesses on top had no effect on the increase in live cells present. Speaking of how the printed cells grew in culture, as cells are cultured, they multiply and grow, and this subsequently increases the mass of the culture. When 3D printed plant cells were grown in maintenance media, they more than doubled their mass, but were spread out and halved in density. while in induction media, the cells increased their mass by around 40% and were more dense and less spread out. The authors next performed a dynamic mechanical analysis, which informs them of how well the cells retain their shape when put under mechanical stress. They found that cells in maintenance media had properties that more resembled the gel they were originally cast from, while cells in the induction media had more wood-like properties. Now, beyond cell wall composition, cellular form can also impact the dehydrated material properties by influencing material microstructure. When cells are printed and not given media to support cell growth and then dehydrated, cells are discrete, flattened structures with no interconnectedness. Naturally grown samples are notably thicker and contain a higher proportion of plant wall material, shown in blue. The stem also shows multiple tissue types with lignin spread throughout, characterized in green. Here the structure is more dense and the vascular xylem tissue is localized to the stem. Now, materials grown in maintenance media yield an open microstructure with rounded cells, which retain their shape on dehydration, and there is low lignification. Materials grown in induction media, however, exhibit a tightly packed network of plant material with a variety of cell configurations, which leads to increased interlocking of the plant material phase, and there is abundant lignification throughout. Now, to quickly summarize everything all together, to start, the authors combined plant cells with hydrogel to create a scaffold and guided cell differentiation with different media types. This was printed into different shapes that developed over time. And when looking at the structural properties of the finished results, the authors identified that variation in hormone levels in cell culture impacts physical properties, microstructure, and mechanical properties. This results in chemically variable materials which can be further utilized. Now, not only do I think that these cellular manipulations are exciting to investigate and learn about, they are also significant in a broader context. This information is significant because the authors show that plant cells can be essentially printed and thus created in different forms. These cells are also not fixed in their properties. Depending on the nutrients they're exposed to, they can differentiate in different ways, allowing for a tunable process to create different material properties. Thus, in theory, we can influence these cells to have properties similar to wood, which would be useful in meeting the growing demand. All science is basically a stepping stone for new knowledge, and these steps are driven by questions. And I had a few questions myself after reviewing this information. The first question I have is whether these cells can be influenced to differentiate in stages, so that they can be left to grow to the desired shape and thickness, and then the media is changed so that they develop useful functional properties. Or is this something that has to be printed in a certain conformation and then only allowed to have one treatment to it? Another question I have revolves around biodegradability. Though the authors are using plant cells, which are biodegradable, does the process of changing the natural fate of the cells maintain their material biodegradableness? Or will products created in this fashion have to have a specialized waste disposal process when they inevitably deteriorate? Though this research is still in the early stages of development, what are the projected costs associated with the final product? Will this actually be a viable alternative to classical forestry? As always, though, my final question revolves around you. What sort of ideas or questions popped into your head when hearing about this information? I would love to hear about them in the comment section below. Also, let me know if there are any topics that you'd like to hear about in the future. Ultimately, I hope that you learned something, but more importantly, I hope you enjoyed doing so. If you did, give this video a like and subscribe for more in the future. Well, that's everything for today. Thank you for watching, and I'll see you next time.